Thank you to ACSM for providing the platform to present research in light of the COVID-19 pandemic. My name is William Gowers, a PhD researcher at the University of Kent in the United Kingdom. On behalf of my colleagues, I'll be presenting our research on the effect of acute beclomethasone and prednisolone on 40km and recovery for subsequent 10km cycling time trial. Athletes with asthma-related conditions are commonly prescribed preventative therapy in the form of glucocorticoids, GC. It remains unclear if GC therapy provides a competitive advantage in single bout exercise performance or recovery for a subsequent bout. Proposed ergogenic action includes physiological, psychological, and metabolic mechanisms. However, there are inconsistent findings within literature. The World Anti-Doping Agency stipulates that GC requires a therapeutic use exemption during competition for oral administration, but not inhaled doses. Therefore, this research aimed to compare the ergogenic action of acute therapeutic oral and supratherapeutic inhaled GC administration on 40km time trial performance and recovery for a subsequent 10km time trial performed on the same day. Six non-asthmatic male cyclists had completed the study at time of analysis, with their characteristics shown in Table 1. Following a familiarization session, in a randomized single-blinded order, participants completed 5 experimental conditions: either 1600 micrograms inhaled beclomethasone, 0.5 milligrams per kilogram prednisolone, placebos of those conditions, or underwent a control where neither a drug nor placebo was administered. The protocol can be seen on the left of the slide. In short, 4 hours following administration, a 40km time trial was completed on a cyclist's cyclogometer. Following 1 hour passive recovery, participants completed a further 10km time trial. Physiological response was recorded continuously during time trials. Capillary blood lactate and glucose were sampled at multiple time points before, during, and after exercise. In addition, subjective overall recovery was reported before and after each protocol. Data was analysed using repeated measure ANOVAs with Bonferroni post-hoc comparisons. Figure A shows the mean and individual completion time for the 40km time trial. No significant difference was seen in completion time between conditions. As you would expect, there was an effect of time on physiological response of heart rate and oxygen uptake, but no condition time interaction at any time point, as shown in figures B and C. Likewise, there was no condition time interaction in perceived muscle pain in figure D. There was no significant difference in completion time for the subsequent 10km time trial, as seen in figure A. Furthermore, subjective overall recovery rating was not different between conditions, seen in figure B. There was also no significant difference in blood lactate at any time point between conditions. As shown in figure D, prednisolone maintained blood glucose concentration at baseline levels during the 40km time trial, whereas all other conditions resulted in reduced blood glucose during exercise, and significantly lower than that of the prednisolone condition. Although only preliminary findings, the present study demonstrates that 2 methods of acute GC administration, including one that requires a therapeutic use exemption, did not improve cycling time trial performance, or perceived recovery between bouts. Although no previous research has investigated 2 administration methods in a single study, these results support previous literature by Kuipers et al. And Hustrup et al. Demonstrating no improvement in exercise performance following inhaled GC administration. However, those investigations featured short-term administration rather than acute doses. Our results are in agreement with Arletes and colleagues, who demonstrated no increase in exercise performance following acute oral GC administration. As in previous literature, the present study demonstrated hyperglycemia following acute oral GC administration, but adds evidence that acute supratherapeutic inhaled administration does not induce this systemic response, nor does this mechanism result in performance enhancement. The wider context of this research indicates that wider guidelines to allow oral GC during competition under the TUE system may be appropriate. Future research should investigate the effect of longer duration administration on arguably more ecologically valid time trial performance, and particularly recovery between endurance exercise bouts, due to GC's powerful anti-inflammatory properties. The present study has collected venous blood samples to investigate inflammatory cytokines between exercise bouts, with analysis due to commence shortly.